Hello, everybody, and welcome to another episode of Scientific Riz. Today, we're going to be covering the phospholipid bilayer and how molecules can travel across it. Phospholipids have a hydrophilic phosphate and glycerol head and two fatty acid hydrophobic tails, which one of them would be unsaturated, which has a break, and another which is saturated, which is in a straight line. Now, the phospholipid bilayer of a membrane is primarily held together by hydrophobic interactions between the fatty acids of the phospholipids, which are repelled by water and naturally aggregate to form the bilayer. The same forces that form phospholipid bilayers cause them to rearrange and spontaneously heal small tears. We're going to see this in action later on. Now, there are two ways that there's transport across a cell. There's active and passive transport. Now, what is the difference between them? Active transport moves molecules against their concentration gradient. In other words, from an area of low to high, which is usually in the form of energy of ATP. Active transport is divided into two types. Primary, which is directly using ATP, and secondary, which, in other words, secondary active transport, when there's a molecule moving down, another one moves up. Now for passive transport moves molecules along their concentration gradient from an area to high to low concentration, and they do not require energy. The fusion is an example of passive transport, where it balances out the concentration gradient between the cell and its external environment. Now, what is facilitated diffusion? Facilitated diffusion requires the use of proteins embedded in the cell membrane when the molecule is large, polar, or charged. There are two types of these proteins. There are carrier proteins, which can do active or passive transport as they bind to molecules and change their own shape, and channel proteins, which only can do passive transport as they allow molecules to passively move along a concentration gradient without using energy. Now, large amounts of water travel across channel proteins called aquaporins. Now, another thing that's important when we're talking about active and passive transport is endocytosis versus exocytosis. During exocytosis, vesicles, which are membrane-bound sacs, expel molecules during active transport. During endocytosis, they do the opposite. There's three types of endocytosis. Pinocytosis, which is known as cell drinking and occurs with smaller molecules. Phagocytosis, which is known as cell eating and is used to engulf larger molecules. And receptor-mediated endocytosis, which can be thought of as a spy, kind of, where receptor proteins embedded in the cell membrane search out specific molecules in order to engulf them. However, in all cases, vesicles do something like this, where they engulf the molecule, form their own layer around the molecules, the cell membrane returns to normal, and they move into the cell. Thank you, and I'll see you next time.